Why do living things look so different? What's happening inside them? What if I told you every cell is constantly talking? Today, we'll learn how to listen. Everything in biology starts with what we can see. Maybe a plant survives drought. Maybe a patient develops a disease. Or maybe two cells look identical but behave very differently. This visible outcome is called the phenotype. But here's the key question. What's happening inside the cell to cause this? Inside every cell, DNA contains all possible instructions. But not all genes are active at the same time. Some genes are turned on. Some are turned off. This process is called gene expression. From DNA, RNA to protein, RNA is like a snapshot of which genes are currently being used. So, if we want to understand phenotype, we need to measure RNA to see which genes are expressed and how much RNA is there. But RNA is fragile and invisible. We use RNA sequencing to capture the full picture of gene expression. So we prepare it for sequencing through several steps. Extract RNA from cells. Convert RNA to cDNA to make it more stable. Add adapters like barcodes. Amplify fragments. This creates an RNA-seq library to turn biological messages into readable digital fragments. Imagine millions of tiny RNA pieces sitting on a glass chip. Now, the machine starts reading them one letter at a time. It adds a glowing building block, a U, G, or C. Each one shines a different color. A camera takes a picture and records the color. Then it washes and repeats again and again, like building a sentence, one letter at a time. Step by step, the full RNA sequence appears. In the end, all those colors are turned into digital code, giving us the exact RNA sequences inside the cell. After sequencing, we get millions of short reads. Now the real challenge begins. Turning raw data into biological insight. Typical steps. Quality control. Align reads to genome. Count reads per gene. Normalize data. Find differentially expressed genes. At this stage, we move from data to information. But numbers alone don't tell a story. We visualize the results. Heat maps, patterns of gene activity. Volcano plots, significant changes. PCA plots, sample relationships. Pathways, biological meaning. Now we can finally answer, which genes explain the phenotype? Transcriptomics is like listening to the cell's conversation. Phenotype is what we see. Transcriptome is what the cell is saying. And RNA-seq is the technology that lets us hear it. From observation, to molecules, to data, to understanding. That's the journey of modern biology.